Hello everyone, welcome back to the session. So we are discussing human reproduction chapter, and in that we were discussing which concept. We are done with gametogenesis and we are also done with the menstrual cycle in the previous class, right everyone? So, and we have also begun fertilization and implantation in the previous class, and I gave you some basics of fertilization. I told there were reactions from the sperm side, okay? You call it as acrosomal reactions. And these are all because of acrolysins that are secreted by the sperm. We have discussed this part, and we have three different types of acrolysins. Can you name them? There is hyaluronidase, which is going to break the hyaluronic acid present in between granulosa cells. Then we have corona penetrating enzyme that is going to break the barriers of corona penetrating cells, corona radiator cells. And then we also have zona lysins. Zona pellucida is a glycoproteinaceous nature. Zona lysins are going to dissolve the zona pellucida. Clear, guys? Yes. Now we are going to discuss about zona and cortical reactions. Did I mention this? Zona and cortical reactions. These reactions happen at the region of ovum, and these reactions are the reason for controlling polyspermy, or else polyspermy can take place. So this checks polyspermy, slow block polyspermy. This will be checking polyspermy, slow block polyspermy. Am I clear with this? Yes, everyone. So now taking zona and this cortical reactions. Guys, if we take this is the secondary oocyte still, and it is going to have a zona pellucida layer like this. And it is also having a corona radiator layer. The cells of corona are this. Like more than one layer of cells are present here. Like this. Okay, now. So with the help of that acrolysins, sperm is entering till here, okay? Now it is here in this perivitelline space. So the space present in between the plasma membrane as well as the zona pellucida is called perivitelline space. Okay, now as soon as the sperm head is touching the plasma membrane of the ovum, ovum layer is going to get depolarized. The plasma membrane of the ovum gets depolarized. The first point, as soon as sperm head touches the plasma membrane of the ovum, yes, the membrane, it is going to get depolarized. There will be influx of sodium. We have already learnt in neural control and coordination chapter how depolarization happens. Influx of sodium causes depolarization. There will be influx of sodium into the secondary oocyte. This causes depolarization of the membrane. Guys, this depolarization, it is not allowing the other sperms to enter inside, and this happens very fast. Depolarization is very fast, and this is the first step to stop polyspermy. This is the first step, and we call this as the fast block polyspermy. This entire thing, this is called as fast block polyspermy, as it is happening very faster. Okay. Now, if you go to the zona and cortical reactions, guys, after this happens, inside the ovum, so there are some granules here, okay, and we call them as cortical granules. There are certain granules called as cortical granules. So this is the second step of slow block polyspermy. So as soon as the sperm head is touching the plasma membrane of ovum, so cortical granules are released into the perivitelline space. These are released into perivitelline space. Perivitelline space is the space in between the zona pellucida as well as the plasma membrane. Okay. So this causes entry of calcium first. Calcium enters inside the ovum, the secondary oocyte. Entry of calcium, this results in release of cortical granules, okay? And these cortical granules are having some chemicals. So they are going to completely block the entry of other sperms. Block the entry of other sperms. How it is blocking the entry of other sperms? By destroying the receptors on the ovum membrane. Destroys the receptors. Destroys the receptors on the secondary oocyte. Clear everyone? On the plasma membrane of the secondary oocyte. Guys, see here, we have something called as, what should I tell? Only human sperm can fuse with a human ovum. Do you know that? The reason is there will be receptors for both. Sperm is going to carry certain chemicals and ovum is going to carry certain chemicals. So fertilizants and anti-fertilizers, this is what we call them. So they are specific for a species. This is why interspecies hybridization is not allowed naturally in nature. Clarified? And the zona pellucida, right, it is having some receptors, not zona pellucida, the plasma membrane of the ovum, it has some receptors. So that's receptors, sperm requires that receptors to enter into the ovum. And these cortical granules that are released, they will destroy the receptors on the secondary oocyte such that second sperm cannot enter the ovum. So here two things. The first thing is fast block polyspermy, that is because of sodium, and the second thing is cortical granules. Because of entry of calcium into the ovum, cortical granules will be released and that cortical granules are destroying the receptors. Am I clear how polyspermy is prevented? Yeah, this is how polyspermy is prevented. Yeah, now we have a concept of sex determination, how the sex of the individual is determined. Sex determination. Okay, can you guys tell me how sex determination is happening? That is depending upon the chromosomal thing. So if we take a female, female produces a single type of egg. Only X type of egg. Out of 43 chromosomes, right? 46. Out of 46, out of 23 pairs of chromosomes, the last set now we call them as what? Sex chromosomes. So in that sex chromosomes, females usually produce only one type of ovum, that is X type of ovum. If you take a male, male can produce two types of sperms, X as well as Y. So imagine this kind of sperm got fused. So the ploidy will be X and X. Imagine this kind of sperm got fused, the ploidy would be X and Y. Understood? If that is a double X ploidy, that is a female offspring. If that is a XY ploidy, that will be a male's offspring. So this is how sex is determined. So sex of the individual is determined by the chromosome that father is having in the sperm. Are we clear with this? Okay, now let us move to the next concept called as embryonic development. We are done with the fertilization and implantation part. Let us go to the concept of embryonic development. Yes, everyone. So this all begins with fertilization, entry of the sperm nucleus into the secondary oocyte. Guys, entry of sperm. As soon as the sperm is touching the plasma membrane of the ovum, secondary oocyte completes meiosis 2. Secondary oocyte, it is going to complete meiosis 2, and now it is turning into a ovum as well as a second polar body. Understood, guys? Understood? That is meiosis 2 completion. So after fertilization or just before fertilization, secondary oocyte completes the meiosis 2. If fertilization is not happening, what will happen to the secondary oocyte? The life of secondary oocyte is only 24 hours in the female reproductive tract. If fertilization does not happen in that 24 hours, that will degenerate in the form of secondary oocyte. It is never going to convert into ovum. So every month, what's going to happen on the day of ovulation? Secondary oocyte is released. If that is not fertilized, what is going to expel out of the uterus? Secondary oocyte is getting expelled out of the uterus. It is not ovum. That's why I'm like stressing this point. Don't call it as a ovum. It is a secondary oocyte before fertilization. Okay. Now this fertilization, it is going to result in what? A diploid zygote. Fertilization results in a diploid zygote. Clear everyone? So during fertilization, both sperm and ovum nucleus, chromosomes are released, and these chromosomes, they form a mitotic spindle. So that is what? A 2N ploidy nerve. N chromosomes from the sperm as well as N chromosomes from the ovum. Now they have formed a mitotic spindle. Now cells will divide through mitosis. Okay, that divisions, now we are calling them as cleavage divisions. So name of the topic is cleavages. Guys, if you take cleavages in animal kingdom, these are very slowest. Okay, if you compare with rest of the organisms, plants and all, especially humans, we do have slowest kind of cleavages. So slowest in animal kingdom. Yeah, now this diploid zygote, it is not going to stay in that stage only. That is just a single cell. Zygote is a single cell, that's it. It is not going to stay in that stage, so it has to divide. The first cleavage of the zygote, this will happen 30 hours after fertilization. So this is the first cleavage that is passing from animal axis or animal pole to the vegetal pole. Animal pole to the vegetal pole, and this is meridional plane, a straight one. This is meridional plane from the center of that ball of zygote. That is meridional plane. It is passing from the center exactly. Okay. So this is going to result in two cells. Now we call them as blastomeres. So cells of the zygote, the first cells of the zygote is called blastomere. Done everyone? Now there will be a third cleavage. Third cleavage is slow and it happens like this. Okay, it is not going to divide the zygote into 4 cells. Instead, third cleavage, I'll write the first cleavage. So first cleavage, this happens 30 hours after fertilization. See, after a day, 30 hours means after a day almost, the first cleavage is happening. And second cleavage, now it will take one more 30 hours. And second cleavage, this is an unequal cleavage. So this results in the formation of 2 cells, 2 equal cells. This is a holoblastic cleavage. So it is divided completely. When the zygote is divided completely, that you call it as holoblastic cleavage. And that is resulting in 2 equal sized blastomeres. So the first division is a holoblastic as well as a equal division. And if you take the second cleavage now, it is resulting in a transient 3-celled stage. Can you see that? Transient means temporary. This is resulting in a transient 3-celled stage. So this is a unequal division. This is a unequal division. Unequal blastomeres. Understood now? Only the first cleavage is a holoblastic cleavage. Then after, there is no axis or plane in which cleavages are happening. So that happens randomly. Like you can't tell, ma'am, that happened in longitudinal plane, that happened in a vertical plane. That's not possible. They happen randomly. Cells divide in unequal sizes. Some of them divide in equal sizes. Then it becomes faster. The first and second cleavages are really slow. After that, they become very fast. Okay. This is the first thing. Please copy it. Yes. So now zygote, it is acquiring cells and that is at the stage of 8 to 16 cells. Okay, so at the stage of 8 to 16 cells, a solid ball of cell, this is a solid mass of cells, and this is called as morula. Whenever there are 8 to 16 cells in an embryo, that embryo is called as morula. Understood now? Guys, morula is having both zona pellucida as well as corona radiata. This is very important. That is morula with a nucleus, and morula is still having the intact zona pellucida. It has zona pellucida around it, that glycoprotein layer, and it is also still having the corona radiata. This is the corona radiata, CR, corona radiata, and the pink one is zona pellucida. Clear? This morula now slowly it is passing through the fallopian tube from ampulla to isthmus. Now it is descending down into the uterus. So it will take 4 to 6 days to descend down into uterus. So it will take 4 to 6 days to descend down into uterus. Okay guys? Yes. Now what is the next stage after morula? Morula is going to become a blastocyst, and now blastocyst will be having 16 to 32 cells. When it is called as a blastocyst, guys, morula, I told this is a solid ball of cells, right? There is no space inside the morula. Only like 8 to 16 cells are present inside a morula, a full solid ball. Now this morula, it has to turn into a blastocyst. Blastocyst where there is a small space. This is zona pellucida again, and this is corona radiata. Yes, the cells of the morula, they will start moving towards a corner. Now here we are having more than 8 cells, more than 16 cells, nearly 32 cells will be present. Okay, yeah, this mass is called embryonal knob. That mass of cells that moved to a corner is called as embryonal knob, and the space here is called as blastocele. That space is called blastocele. Clear guys? Whenever it is having this blastocele, then it is called as a blastocyst. When it has blastocele, we call it as blastocyst. That is a 16 to 32 celled stage. Am I clear till here? The second phase. Now blastocyst is ready for implantation. It has to attach to the wall of the uterus. Which wall? It has to attach to the inner layer of the uterus, that is endometrium. Yes, if we take uterus, that becomes the endometrium of uterus. Blastocyst has to now come and it has to attach to where? Yeah, this is blastocyst. It is attaching to the endometrium. This region is endometrial lining of the uterus. Understood? Yes. Guys, before attaching, it has to undergo some changes. So it has to break the zona pellucida and it has to come outside, blastocyst. So before implantation, blastocyst breaks the zona pellucida. Because of the cells now, it has become like packed kind of structure. Zona pellucida is a non-cellular layer, so it is not allowing the further growth, and the mass of the cells now, it became like too solid, and the zona pellucida will start breaking off and blastocyst will be liberated out of the zona pellucida. So blastocyst breaks down zona pellucida, ZP, and implants into the wall of the uterus. This implants into the endometrium. Are we clear with this point? So before implantation, zona pellucida has to get lost. Guys, whenever blastocyst is breaking zona pellucida and it is liberating itself out, there is a chance the embryo itself can get broken down into two pieces. blastocyst itself can get broken down into two pieces. So at that times now, how many embryos are there now? Guys, this ball of cells now, they are totipotent cells. Okay, they can give rise to any kind of cells in the body. Those are totipotent cells, the blastocyst cells. So whenever the blastocyst is breaking into two pieces, while this liberation, two embryos will be formed, and those are identical embryos. That is how identical twins are formed. Understood? Like I myself, I have non-identical twins. My baby boys are non-identical, like one resembles me and one resembles their father. So non-identical twins case na, two ovums and two different sperms. Understood? Two different ovums. A woman has to produce two different ovums in a single cycle of ovulation, and that two ovums will be fertilized by two separate sperms. That results in formation of a non-identical twin. Identical twins, they are formed from the same mass of cells. As they are formed from same mass of cells, they will be same, like as it is, same nose, same mouth, same everything. And don't imagine in this cinematic story, ma'am, identical twins na, if one is crying, the other cries. If something happens here na, something happens there to that twin. That case is not there. Okay, but physically they are same and they share the same genetic material. So as they share same genetic material, if there are any genetic diseases, so that genetic diseases will be seen in both of the offsprings. Okay. Now here starts implantation. Are we clear with this? There should be some image because I can't draw too many diagrams. Guys, I'm showing this image. See. So till here I have explained you. This is morula. Day 2 cleavages. You can see. Day 1 fertilization. The cells will divide and slowly like they are differentiating. Guys, this is the stage of blastocyst. Slowly. Okay, now blastocyst, it has acquired a space inside. I told this is the inner cell mass, right? Guys, inner cell mass, that is made up of cuboidal cells. And now the outer cells, this outer cells, they become flat and we call them as trophoblast cells. You want me to write it? Shall I take another board? This is placenta, no? Okay, I'll erase this part and I'll continue here, okay? Yes. So now where are we? We are at the stage of blastocyst. Now blastocyst do not have zona pellucida. It does not have corona radiata. Both of the layers are gone. Okay. So this is a simply inner mass of cells and the outer mass of flat cells. The outer cells, they become flatter. And inner mass of cells, they move to one region like this. They move to a corner region like this. Right, everyone? So the inner mass and you have the outer mass. Outer mass of cells, that flat cells, we call them as trophoblast cells. These are the cells of trophoblast. Yes, now this trophoblast cells, that will start giving villi. And that villi, they will pierce the wall of the uterus. These are the endometrial cells. Okay. These are the cells of the endometrium. I have taken a small part of endometrium. What happens is, yes, this is the trophoblast cells. The cells of the trophoblast. They will start producing villi like this. And this villi, they will pierce into the endometrium. Yeah, this is how it is formed. And this villi are called chorionic villi. They got interdigitated into what? Endometrium of the uterus. Like tree, it is going to give branches. In the similar way, embryo is going to give branches to fix itself into the wall of the uterus. Clear, guys? Yes, now what's going to happen? The inner mass of cells, it will start forming layers. The inner mass of cells, it will start forming layers, like you do have two layers. One will be cuboidal cell layer and the other will be columnar cell layer. The bottom one becomes cuboidal and the outer one becomes columnar cell layer. Now we are giving naming to this. These cells, they are the cells of epiblast, and the bottom cells, these are the cells of hypoblast. And there is a cavity here. That cavity is the future amniotic cavity. This cavity here, this becomes the amniotic cavity, where amniotic fluid will be secreted as the embryo is growing into stages. Till here you understood? Now the next stage is called gastrulation. The next stage after implantation is called gastrulation. Guys, this inner cell mass, okay, if you look from the top view, that is like this, your palm, isn't it? What you are seeing here is the side view only. From the top view, now it will be like this, a layer of cells. So that layer of cells, we are just cutting the layer of cells. So you have this upper columnar cells. Yes, in this upper columnar cells, what happens is there will be a invagination formed. Okay, that is a cavity. That is a cavity being formed. And we also have this lower mass of cuboidal cells. So these are the lower mass of cuboidal cells. So that is epiblast and the bottom one is hypoblast. Hope that's clear for you. Epiblast and hypoblast. So this is a groove. You call it as primitive pit, or like it has different names. That is a small groove like structure. So into that groove, what happens is this epiblast cells, they migrate. Epiblast cells, they migrate and they enter, they fill this space. They push the hypoblast cells down. They are forming a new layer of cells. So this first formed cells are which layer? These are the endodermal cells. So endodermal cells are the first cells to be formed in a embryo. Next cells will be the mesodermal cells. This is where the mesoderm is developing. Am I clear now? Did you understand from where all the cells came? From the epiblast cells. And finally, the epiblast cells will mature and they are going to turn into ectodermal cells. Now understood from where embryonic layers are formed? This is how embryonic layers are formed. A hypoblast is replaced by endodermal cells. Above endodermal cells, mesodermal cells will be formed and the epidermal cells are turning into ectodermal cells. So now see, understand the source for all of the embryonic layers is which layer? Epiblast. Epiblast is the source for all of the future embryonic layers, and hypoblast got replaced by proper endodermal cells. So this is hypoblast, a primitive endoderm, and this is epiblast, a primitive ectoderm. So epiblast, this is the source of all embryonic layers. This is the source of all embryonic layers. Am I clear, everyone? This part is called as gastrulation, and from here organogenesis will begin. So from ectoderm, nervous tissue will be formed. From mesoderm, what is being formed? Connective tissue and muscular tissue. And endoderm results in the formation of glands of the body. Ectoderm, mesoderm, endoderm, together they form epithelial tissues. So in formation of epithelial tissues, all of the three layers are involved. So that is the origin of the entire human body. Understood now? Clear? Okay, you can compare from this picture also. See this picture. So this is a laminar disc. The zona hatched implantation into the uterine epithelium, that is uterine wall implantation. Epiblast, hypoblast and trophoblast, this is amniotic cavity. Then they got rearranged into laminar disc. So in that disc you can see there is a primitive streak. I told na, I wrote it as groove. So through that primitive streak na, cells are migrating and they are completely forming a 3 different layered embryo. So here is amniotic sac, this is trophoectoderm that forms the placental villi. These are the placental villi, and here is the mesoderm, that is embryo being formed, and slowly the body organs will start forming. That much is not needed because extra embryonic layers, chorion, amnion as well as yolk sac, these are called as extra embryonic layers. You need not to dig that much into it, like how chorion is formed, which cells are forming the chorion. They are from the trophoblast cells actually. I gave trophoblast cells. Trophoblast cells are forming extra embryonic layers. Okay. So I hope you have copied this. Yeah, now moving to placenta. So guys, how placenta is formed? Placenta is formed by the trophoblast cells formed from the trophoblast cells as well as the endometrium. That is a fusion of both the layers, formed from trophoblast cells and endometrium. Trophoblast is from the embryo and endometrium is the layer of the uterus. So half layers of placenta are from the embryo and the half layers of placenta are from mother. Okay. So what should I tell is this is a connection between mother and fetus. This is connection between mother and fetus. Yes, what can pass through placenta? We have already come across this part so many times. Guys, can blood cells pass through placenta? Placenta is a barrier. So that allows only antibodies, not all antibodies, smaller size antibodies, antibodies against smallpox, antibodies against diphtheria and measles. Now they can transfer from mother's body to fetal body through placenta. So that is giving some kind of immunity to the fetus. Okay, so few types of antibodies, nutrition and hormones also, as well as gases and excretory waste. Gases and excretory waste like ammonia related things. So this can pass through the placenta. And placenta is also called as decidua. It has another name. We call it as decidua. The meaning of decidua is falling off. Guys, we do have different types of placentas. In some animals, placenta will be detached along with the fetus. Delivery of fetus happens. After few minutes, placenta will be also automatically delivered. It will get detached. And in few species of animals, only like small part of placenta can get detached, and the other part it will get degenerated. We call it as absorption. It will get absorbed or degenerated within the uterus itself. In case of humans, that is a deciduous placenta. It will be removed after the parturition. Clear? And placenta is a source of so many hormones also. It can secrete thyrotropins. The major hormone is HCG. Guys, HCG hormone is secreted from the trophoblast cells. So that is a hormone from the embryonic cells, not from the mother's body. And this HCG is excreted through urine. HCG is excreted through urine. So if HCG levels are recorded in the urine, what does it mean? So this gives a positive pregnancy test. This gives a positive pregnancy test. And this test is called as Gravidex test. It has another name called as Gravidex test. Clear? So apart from HCG, it is also secreting so many hormones, like it secretes Thyrotropin. See, thyrotropin is a thyroid related hormone. Fetus has to develop. Differentiation of the cells has to happen. If differentiation of cells is not happening, cells will remain in the same stage. Epiblast cells will remain like epiblast. Hypoblast will remain like hypoblast. We come across differentiation, redifferentiation, dedifferentiation terms in botany. So for that we require thyroid hormone for metamorphosis. So thyrotropin, corticotropins. What are corticoid hormones? Hormones from the adrenal cortex. So they are also secreted from the placenta. And we also have somatomammotropin. Somatomammotropin, this is placental lactogen. This helps in production of milk by the mother. So this is stimulating the milk production. We call it as placental lactogen. Yeah, these are some of the hormones of the placenta. Yeah, next moving to the developmental stages of the fetus. Yeah, pregnancy. Guys, an average pregnancy is lasting for 38 weeks, okay? In humans. In case of cat, it will be like 60 to 65 days. In case of elephant, it is more than 350 days. And in case of rat, it is approximately like 30 to 35 days. So from animal to animal, so the duration of the pregnancy, what we call as gestation, will also change. And before that, we have to do the embryonic development. Guys, if you take embryonic development, human pregnancy is of 38 weeks, I told. Or you can roughly call it as 9 months also. A full term pregnancy is full of 9 months. So here in the first month of pregnancy, what is formed? First month of pregnancy, a fetal heart is formed. So heart is the first organ to be formed. Okay, that is where the doctor is going to check the heartbeat of the fetus. If it is not going to develop a heart, then it is going to get eliminated out of the body. That results in a miscarriage. So first is the pumping machine. Blood has to be pumped for the fetus to survive. So fetal heart will be formed. By the end of the second month, limbs and digit. Actually, you don't call them as limbs. You should be calling them as limb buds. Guys, limbs are full hands and full legs, isn't it? Limb bud means tiny projections will appear here and here. So budding like structures will see, bud like structures will be seen. You call them as limb buds. And like tiny digits will also start forming from the limb buds itself. Digits meaning is fingers. Okay. By the end of the third month, major organ systems like excretory system begin to form. They are beginning to form. Major organ systems are formed. And by the end of the fourth month, the first fetal movements can be seen. Fetus movements can be felt. It's not seen. Fetus will start moving as soon as it is formed. Like the limbs and digits are formed, it can start moving inside, but the size of the fetus is too small. Mother cannot feel it externally. For the mother to feel it, at least it should be little bigger. So by the end of the fourth month, it becomes little bigger. Hands and legs will be developed, so that movements can be seen on the uterus. So if uterus feels the movements, she can feel the movements inside the abdomen. So first fetal movement are felt. First fetal movements are felt. Okay. By the end of the fifth month, so you can see like hair will be formed, and by the end of the sixth month, we can see lanugo will be formed. Eyelids are separate actually. Lanugo is the hair on the body. We are mammals now. Mammals will have body hair as a feature. So body hair will be formed, and eyelids, they separate. Like these are the events happening in the first trimester. And by the end of the second trimester, by the end of ninth month, after this now, there is no new organs being formed, but the fetus has to grow in size. It is very smaller in size. It has to acquire full fat, or else it cannot survive in this environment. So there should be a lot of body fat to be accumulated. So by the end of 9th month, it is fully developed. And fetuses born before like 38 weeks, now they are underdeveloped fetuses. Their body systems are formed, but they are not completely matured. So immature birth, now they require NICU attention. Clear everyone? Yes. Now we'll be moving to a concept called as parturition. Yeah, what is parturition and lactation? Guys, expulsion of a fully developed fetus is called parturition. Expulsion of a fully developed fetus. And this is a neuroendocrinal mechanism. Nervous system is involved as well as hormonal system is also involved. Parturition is a neuroendocrinal mechanism. Yes. So from where the signals of parturition are starting? So the signals for parturition, they are starting from a fully developed fetus. Signals arise from a fully developed fetus as well as placenta. A fully developed fetus, what it is doing, it is not telling anything to the brain. The thing is, it is exerting a lot of pressure on the walls of the uterus. A baby is too large and the uterus will get overstretched than its limits. Almost like the volume will be increased by like 400 times by the end of the 9th month. So that extended walls, the myometrial walls of the uterus, they are overstretched. As they are overstretched, they keep sending impulses to the brain. So please expel the fetus out of the body. It is fully developed. So that is what. And placenta is also secreting a hormone called as relaxin. Guys, relaxin is a hormone that relaxes pelvic ligaments. So fetal expulsion can happen. Without the relaxation of pelvic ligaments, fetal expulsion will not happen. Okay. Yes. This fully developed fetus and placenta, they are putting pressure on the uterine walls. So brain is sensing them, and what brain does? Brain is releasing oxytocin. Brain releases oxytocin. As we all knew, oxytocin is a hormone that increases the contraction. So this increases contraction, and this increased contractions, they still send signals to brain. Muscles are too much stressed, and again they send signals to brain. Brain in turn releases more and more oxytocin. So this is a cyclic process. This continues till the parturition is completed. Okay. Yeah, 20 minutes after parturition, placenta will be also delivered. That's why I told that is a deciduous placenta. It will not stay inside. Placenta is expelled out on its own. Placenta will get detached from the endometrium. Yeah, with this we are done with the parturition mechanism. Right everyone? In the next session, I'll have a small part of lactation. We almost discussed about milk production. So in the next class, we will discuss about lactation, and we are going to end the chapter in the next session. Yeah, see you everyone.